Welcome back to Dining Lab. Today we'll be discussing parts of 2.6 and 2.7 about the structure of DNA and DNA replication. This is a DNA molecule. DNA molecules are made up of deoxyribose sugars, phosphates, and a base. The four bases in DNA are adenine, thymine, cytosine, and guanine. These four bases contain nitrogen and has either one or two rings of atoms in their structure. The phosphate of one nucleotide and the pentose sugar of another nucleotide is linked together by covalent bonds to link nucleotides together into a chain. This creates a strong sugar-phosphate backbone. The bases are linked to each sugar and they pair together by complementary base pairing. Only one purine and one pyrimidine can fit between the sugar-phosphate backbone. A purine has two rings in its structure and a pyrimidine only has one ring. The purine and pyrimidine is linked together by hydrogen bonds. Adenine can only be paired up with thymine and form two hydrogen bonds, while guanine can only pair up with cytosine and form three hydrogen bonds. The 5' terminal is where a phosphate is linked to a carbon atom 5 of the deoxyribose sugar. On the other hand, the 3' terminal is where a hydroxyl group is attached to the carbon atom 3 of the deoxyribose sugar. The two strands that make up the DNA have their 5' and 3' terminals at opposite ends, which means that they are antiparallel. It is important to note that DNA replication can only occur in one direction from the 5' to 3' terminal. Before replication can occur, the enzymes Helicase and Gyrase work together to separate the two strands. Helicase unwinds the double helix and breaks the hydrogen bonds that links them. DNA gyrase moves in advance of helicase and relieves strains in the DNA molecule to ensure that the strands would not form tight supercoils. Because both strands in the DNA run in opposite directions, they require different methods in order to be able to replicate the strands. The leading strand is continuous because DNA polymerase can move in the same direction as the replication fork. On the other hand, the lagging strand is discontinuous because DNA polymerases have to move in the opposite direction to the replication fork. In the leading strand, a DNA primase adds a short length of RNA to the template strand. This acts as a primer which marks the start of the replication. Then, DNA polymerase III starts a replication next to the RNA primer and adds nucleotides in a 5' to 3' direction. The nucleotides are added via complementary base pairing in a 5' to 3' direction. The 5' to 3' direction means that the phosphate group of one nucleotide, which marks the 5' terminal, is added to a pentose sugar on the DNA strand which marks the 3' terminal. In the lagging strand, replication occurs away from the replication fork. So a DNA primase is added closer to the replication fork and the replication starts from the 5' to 3' direction. Primers are added constantly along the lagging strand and short lengths of DNA are formed between the primers, which are called Okazaki fragments. Then DNA polymerase I removes the RNA primers and replaces it with DNA. Finally, DNA ligase seals up nicks in the sugar-phosphate backbone by connecting it with a sugar-phosphate bond. The double helix rewinds, and this results in two DNA molecules. DNA replication is referred to as semi-conservative because both strands are composed of an original strand and a newly synthesized strand. In eukaryotic organisms, DNA is packaged into structures called nucleosomes. Nucleosomes contain 8 histone proteins, which are 2 copies of 4 types of histone proteins. The DNA wraps around these 8 histone proteins, and a short section of DNA connects one nucleosome to the next nucleosome. Nucleosomes are essential in the process of supercoiling to form chromatin from DNA molecules. This allows DNA to be packed into a smaller space within the nucleus. Prokaryotic DNA are not wrapped around histones, so they are referred to as naked. Watson and Crick discovered the structure of the DNA through model making. So they found out that DNA strands are antiparallel and form a double helix. The DNA strands pair via complementary base pairing, and edges of the base enable replication and transcriptional proteins to access it. Before the helical structure, the original model consisted of a triple helix. However, this original model was rejected by Rosalind Franklin's X-ray crystallography data, which showed X-ray diffraction patterns that confirmed the double helix structure. The crystallography data was shared without her knowledge or permission, but now she is recognized as a huge contributor to the DNA structure. The original triple helix created by Watson and Crick also didn't take into consideration Erwin Chargaff's findings regarding the equal number of purines and pyrimidines that should fit within the sugar phosphate backbone. With previous experimental discoveries, Watson and Crick were able to create a new DNA structure, which was the antiparallel double helix. Before DNA was proven to be semi-conservative, there were 3 different hypotheses, which proposed that DNA was conservative, semi-conservative, or dispersive. The conservative model suggested that the old DNA is not altered at all and it results in the creation of an entirely new molecule. On the other hand, the semi-conservative model suggested that each new molecule consists of one old template strand and one newly synthesized strand. In the dispersive model, all of the molecules are made up of segments of new and old DNA. Meselson and Stahl used isotopes of nitrogen, which are N15 and N14, to test the DNA replication models. During the experiment, they first cultured the bacterium E. coli in a medium where the only nitrogen source was N15, so all of the bases of the DNA in the bacteria consisted of N15. Then they transferred the bacteria with a medium that consisted of N14 and collected samples from it, which was separated via cesium chloride density gradient centrifugation. The results showed that after the first replication, the DNA molecules contained a mixture of N15 and N14, which disproved the conservative model, and after 2 divisions, some of the DNA consisted fully of N14.